From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on April 3rd, 2025. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 86th episode of Matters Microbial. I am truly grateful for every download, listen, and view on YouTube. Thank you for spreading the good microbial word far and wide. This podcast is my chance to promote microbiological literacy, and that is a very big deal to me. Last week, our podcast guest, Dr. Rosie Allegado, briefly mentioned one of her research interests, the wonderful choanoflagellates, an early ancestor of you and me. My wife, Dr. Jennifer Quinn, 3D printed this glowing model of a choanoflagellate. At the time of this recording, it is the day after my late mother's birthday. When she was so ill, I tried to keep her mind off her disease. Since she loved to sew and make clothing, I had her make me microbial vests where the material represented microbes. Here is a great example of my mother's work and a close-up of the detail. I will put a link in the show notes about this topic. Miss you, Mom. In the hall outside my little laboratory, I have a whiteboard that urges passerby to jot down their favorite microbe. I've had lots of unusual answers, but this one, bioluminescent thermophiles, made me smile. Then I thought, has anyone looked for them? Maybe I should. Last week, I showed viewers how some bacterial isolates I have found possess antimicrobial activities. But sometimes I see more than that, and this ability is critical for students. Here's a photo of one such isolate on a background of Bacillus subtilis. It looks pretty normal after extended incubation. But look at that same isolate on a background of E. coli and a background of Acinetobacter. You can see on those backgrounds almost a flowery outgrowth. I cannot tell which member of each arrangement is forming the flowing pattern, but it's interesting. Many times, bacteria do strange and wonderful things when in the presence of other microbes. Sometimes we forget that bacteria do not grow by themselves in pure culture. They interact. I adore having students notice this kind of thing, what's often called socio-microbiology. During several prior episodes of this podcast, we've chatted with guests about the primacy of the microbial world. First evolved, last extinct. How they can be found in almost any environment with liquid water and even a tiny energy gradient that they can tap. That, of course, makes me think about life on other planets. But our own planet has some very unusual environments as well that are populated by microbes. So I am beyond honored and grateful to welcome Dr. Karen Lloyd, Professor of Earth Science at the University of Southern California, to the podcast to discuss her upcoming book about Intraterrestrials, microbial life seemingly everywhere, especially deep underground. Welcome to the Quality Quorum today, Karen. It's truly an honor.
Thanks for having me. It's great to be here.
I'm so pleased to have you here. And I want to say something to listeners and viewers to start with. Many times, people think of the idea of a microbiologist sequestered in a laboratory. And I know how that feels because I've been there. But when I was in graduate school, I remember the ecologist Paul Ehrlich talking about the difference between ecology and microbiology. And he showed a slide of Charlie Yanofsky peering into a fermenter. And he said, here's your typical molecular biologist in his lab. And then he shows the Rocky Mountains and says, and here's my lab. And everyone laughed. But I want listeners and viewers to know that microbial ecologists can go a lot of places. And your book demonstrates that.
Yeah, for sure.
And I think a good place to start, if you don't mind, is that term intraterrestrial.
Yeah, I didn't make it up. I first ran into it when the late Katrina Edwards, used it. Yeah, she passed away a few years ago, but she did so much for the field of deep subsurface microbiology, sort of garnering NSF funding to launch all of our early careers. It certainly helped me get a leg up. But she used that in a paper, and I thought it was just such an apt word. And so then when I was writing this book, I was like, well, who do I really credit with this? Like, where did it come from? The first mention that I could find of it was Karsten Peterson, published a paper earlier than that, and he used the word. So I think it was him. But Dave Valentine also used it in a commentary that he wrote on a paper that we wrote. Anyway, that's the background of it, but I just like it because it talks about life inside Earth, and it gives that comparison to life outside Earth. And it's just, I think it's apt.
I think it's interesting because, there's even a Wikipedia page on the term, by the way.
Oh, I didn't know.
Yeah, well, I just love the word myself, so I get curious. That's my thing. But I'd like to quote the jacket copy on your book. And the first thing that I want you to do is tell everybody, and we're going to repeat this, when this book becomes available, because it is an official Matters Microbial must read.
Okay. May 13th of 2025, it's going to be available. But you can pre-order it now if you want to on Amazon, or Princeton University Press is the publisher. So
you should, because it helps with the metrics. And I am thankful that Princeton University Press sent me a pre-publication edition that I was able to read. Yeah. And I want to quote the jacket copy because this is just amazing. You must feel really good about this, Karen. Obviously, Intraterrestrials, discovering the strangest life on Earth, and then the PR stuff. And I don't know if you wrote this, but I love this. A biologist's firsthand account of the hunt for life beneath Earth's surface and how new discoveries are challenging our most basic assumptions about the nature of life on Earth. And that's just a wonderful bit.
Yeah. I mean, I think it's true. I think it's not an exaggeration.
No.
What I know about biology now is very different than what I knew when I was in college.
And I want to point out about this book that it's not just what's under the ground, though that is a lot of it. And so once again, I've always been interested in unusual things. I mean, that's what really puts a sparkle in my eye, a microbial sparkle, for lack of a better term. That's not just a biofilm. But the idea is here that you're touching on all the things that I think are super important for everyone to know. And I want to emphasize with this book, and kudos to you, it's written in a very personable, straightforward, interesting way, almost like a travelogue. And I really appreciated that.
Yeah.
Yeah.
My fear with this book was that it would feel disjointed, that it would feel like, okay, now it's like an adventure story of like racing in trucks across the Andes, and, where are we going to sleep? How are we going to deal with this altitude that's making our heads feel like they're going to implode? And then, and now we're going to talk about the proton motive force. And like, whoa, whiplash. But the reason that I did it that way was intentional, because that is how we live our lives. Those of us who do this work are bopping between, interpreting what kind of respiratory pathway a potential organism has and maybe finding out ways to test that empirically, to, oh crap, if I don't make this flight with my dry shipper, which is this like horrid looking thing that looks like a bomb that we have to somehow convince airlines. It's FAA approved. It's completely legal, but the front gate people, so then, we move seamlessly between these two worlds. So I thought that the book should reflect that.
And I think it's a good balance. And I don't mean to make light of exciting travels. I've read a lot of those books as well. But what I like is there's some actual really solid layperson-based explanations of things that are really worth everyone's time. This is especially important to me in an era where maybe science isn't as respected as it should be. This is a great opportunity. And I would also like to add is so many people have preconceived notions about what microbiology is and what microbiologists do. And so that's why I was so appreciative of it. I think a good place to move on, if you wouldn't mind, is you just came back from a wonderful place.
Yeah.
And I would like you to talk a little bit about the trip from which you just returned, what you did there, why you were interested, and what the next steps are.
So I spent the last 2 weeks in New Zealand going around the North Island sampling hot springs. And this is a collaboration with colleagues from around the world at the University of Naples, at Woods Hole Oceanographic Institute, at universities in Chile. It's really just a big international effort. And so we use hot springs as sampling ports for the subsurface, just because it's an ridiculously huge, impossible amount of money if we just like dug boreholes all over the world to try to get at the subsurface, some kind of landscape scale. You just can't do it. You can't like go to a country and be like, can I poke holes in your whole country? No. Well, the holes are already poked by fracture zones. So these are like active margins. And so the plates are causing things to move, and voids and cracks are created. And so every time that happens, the waters will basically eject the deep subsurface microbes up to the surface. And we do a lot of work to look at the geochemical content to make sure that it is truly from the deep, that it's not, yesterday's rainwater that somehow is popping back up. And we also take samples of the surrounding soils to sort of blank out those. But it was a wonderful trip. We averaged about 2 sites a day, which is pretty good.
And from what I understood, you were in the Rotorua area, which is where you see an awful lot of Maori culture.
Yeah, it was awesome. That is like probably the best part. I've done this type of sampling in a lot of places, but the thing that made the New Zealand work unique was the Maori participation in what we were doing. I mean, the reception that we got was so cool. They are so careful at being guardians of their hot springs, and they've maintained the spring health. It's because of the work and the effort that they've put in to maintaining their culture, maintaining their stories, maintaining all this stuff for 700 years, that I can now arrive on a plane and get these great samples. So like, I want to acknowledge that when I get samples from places like this. But it was just a very cool cultural exchange.
No, I think it is. And, it's really interesting. Every place, and I haven't been to as many places as you are. When I was at Yellowstone, I thought about some of this. Yeah. And actually, when I was in Iceland, I got very interested in the hot water pipes because they're really hot, Karen.
Yeah, I know.
My first thought is what's living in a biofilm inside those pipes. And my wife, Jenny Quinn, said, that's peak Doc Martin. And yes, it was.
Yeah.
So I've been to some of the hot springs up around Rotorua, and they're just wonderful, as you say. And again, I want to emphasize that microbiology is a global pursuit. We're all part of the same biosphere. And it's delightful to hear that the people who have lived in this area for about 1,000 years are interested in supporting the ideas that you're looking at.
Yeah. It's cool that they see the benefits to themselves, and that it feels like a collaboration, which is really cool. And of course, I was working with New Zealand scientists as well, not just the Maori. Matt Stott and Cindy Werner were amazing.
So just for listeners and viewers, a lot of organisms that you find in hot springs are thermophiles or hyperthermophiles, which are adapted to live at higher temperatures. What do we know about life under the earth with regard to that? Is it also so warm, or is it only due to certain areas?
It's not all hot. Some places the thermocline to like 100 degrees is maybe kilometers deep. So you can actually have cool life living pretty deep inside Earth's crust. I mean, at some point, everything gets hot. Like the mantle is universally hot and too hot for life at some point.
Yes.
But we have the full range of temperatures extending into frozen. So the permafrost is a great subsurface environment.
So this is really interesting. So you're collecting these hot spring samples to look for, I presume, thermophiles, or just doing a census of what's there?
We say hot springs because that's what people are used to, but we actually take cold springs as well. Any natural spring. So it's just about the subsurface. It's really not about thermophiles.
Oh, that's so interesting. And you're looking to see what kinds of life is there?
Yeah. So I primarily use nucleic acids to go for these things. So I tend not to grow things so much, although sometimes I do. Mostly because my approach is to catch them in the wild doing what they do with as little intervention from myself as possible. Like I think of myself as like hiding in like a little duck blind, like trying to observe them doing what they're doing. And so that's why I catch them and kill them and, and take all their biomolecules.
Well, without that part, that makes a heck of a cartoon in my mind about the microbial duck blind. I got to get someone to do that for you because that's an excellent image. And I want to point out something that we've touched on in earlier podcasts is that cultivation of microbes is a form of selection.
It is.
And one of the, and I see it in my own lab, one of the problems that we have is we don't know what the originals were. So for everybody that says, oh, you should be able to grow these things, I have two answers. The first is you don't know what they need all the time. Sometimes they need one another, as I mentioned earlier, or that you're going to lose the initial form. When I learned about microbial ecology back when dinosaurs ruled the earth, Karen, it was all about cultivation. And at the time, I wondered about that. Does that really reflect that's there? But you get a snapshot with the nucleic acids.
It's amazing that we've been able to get as much as we have from cultivation, given what a selecting factor it is to say, I want you to grow under my laboratory conditions exenically with no other friends around. And it's actually pretty impressive that we've gotten such an amazing array of things from just that selection pressure. But, even ones that you've gotten into culture, it's kind of like if you wanted to study lion ecology and you just go to the zoo every day. And so then you write a whole paper about how lions' diet is carefully cut up slices of meat. Well, we know that they chase down zebras on the savannah, but that's because we go to the savannah and look at them. It's the same thing with microbes. They may do things in lab that they don't actually do in a certain situation in nature.
Absolutely. There's a wonderful example, and it's very simplistic. I hope you'll forgive me. And I don't have the details of it, but a friend who worked on this sent it to me many years ago. I'll see if I can find the image and we'll post it. And it's basically where you have a micro colony of one type of bacterium here, one on the left and one on the right, and they're all interacting in terms of pigmentation and growth.
Cool.
But even that is on a petri dish.
Yeah.
With cultivatable organisms, right?
Yeah.
So this is really critical that you're looking with a snapshot. What kinds of things are you looking for? Again, some folks just ask who's there, but you're actually able to approach what they're doing.
Yeah. And that's, it's a lot of inference. So you have to hold your conclusions very loosely and be willing to accept alternative explanations. I'm always doing that. But we go deeper into their genomes, of course, with metagenomics and, the work that Jill Banfield has done in the early 2000s to basically crack open our ability to put genomes together from these metagenomes has been a game changer. And so we can reconstruct these genomes and put together genetic pathways. Of course, genes don't mean that a particular process is being expressed and functioning. So we also look at messenger RNA to try to get at whether it's being expressed. And then we look at proteins to see if we can see the proteins are there. But then we also design assays for whatever function we're predicting and apply that to the bulk fluids and then see if we can see that what the functions that we're predicting are actually happening in nature.
It's interesting in the process of many parts of your book, what you're really talking about in some degree is geomicrobiology, which many folks haven't heard of. And I want listeners and viewers to understand that. I talk about something, and I'm sorry if you've heard this from me in some other context, what I call the three deadly centrisms: eukaryocentrism, oxycentrism, and colicentrism. And eukaryocentrism is putting all of our focus on eukaryotes, and that's the way we teach things, unfortunately. I wish we had a totally microbially based curriculum. That would make me happy.
Can I suggest a fourth?
Sure.
Time centrism?
Yes. Oh no, I like that because you reference that in your book, and I want to talk about that at some point because that is wonderful. Because again, people make assumptions. They all grow very fast, and I look at my old notes, And I want to call bacteria are selected, that they're growing as fast as they can, but that's not true.
Not always.
Yeah, there are some that do, and those are the ones that we study because those are the mistakes we make.
And we talk to each other all the time about optimal growth conditions. I mean, that's like one of our basics. Okay, optimal. We use that word. What does optimal mean? Optimal means fastest growth.
Mm-hmm.
Like, it doesn't really mean when we say, oh, I isolated this organism and it grows best at pH 7 and 35 ppt salinity. That meant that it had the lowest doubling time. That may not be the best strategy for that organism. It may be most successful in an environment that has 32 ppt salinity because it's limited a little bit in its growth rate, and that allows it to express some other appendage that has an evolutionary advantage. So just the notion that fast growth is king is, I think, short-sighted.
I agree. And it's funny because that's the way that you're taught. You know,
Always. Yeah.
Single colonies, pure culture, none of these things are true. But to be fair, and I want to make sure I say this, that approach, that reductionist approach, worked very well for human-related microbiology during the 19th and early 20th centuries.
It still
we solved the well, of course,
and E. coli is still useful too.
No, exactly so. I am not disagreeing with you, but I have this theme that we have kind of a image in our heads. I call them devil microbes, that they're always bad, which is not true.
Mm-hmm.
Angel microbes, that they're always good, which is also not true. But actually the third, and I hope you'll like this, I call them meh microbes because they don't care about us at all.
Maybe.
Or we're not smart enough.
Have you spent much time cogitating the fact that there's an entire domain of microbes that causes no pathogenesis?
Yeah. And
there's no archaeal pathogen. Why?
So this is really interesting. Archaea, the third domain of life, fascinating subject, and it's even more confusing than it appears because they easily enter into symbiosis.
Yeah.
Or excuse me, I'm not using that term correctly. They enter into mutualisms quite easily. Okay, within many.
Other than the other branches.
I wouldn't say that, but what I would say is that many people found that they were certainly living in the cytoplasm of protists, certainly methanogens intracellularly, things of that nature. But the reason I bring this up is why no pathogens? And I asked that question when I took the microbial diversity course so many years ago, and I will not name the famous scientist who said, and I quote, "Archaea are vegan. They don't eat meat."
But why?
I know. And that's it. And see, here's the issue. A lot of folks, and I see this in the classroom, they want there to be really exciting answers to questions. But the fact is we know so very little, but we think we know a lot. Well, I don't. And I think what I don't know is exciting.
Yeah.
Because that's an opportunity.
I love a big question.
Yeah. So again, the idea of why archaea tend not to have pathogenic interactions. Some people might say, well, we're not looking hard enough, and that can happen.
Could be. But you say tend not to. I think we can go even further and say absolutely don't, as far as we know. I think it's never been discovered. I could be wrong, but I think that's true.
There's one suggestion that they might be involved with periodontal disease, but that's part of a multi-microbe kind of infection. Yeah. So I
saw that people went kind of hard into that, and they didn't have a pathogenic island. They didn't have any normal virulence factors. They just look like they're, the little sister who gets dragged along in the getaway car.
Or they're taking advantage of the damaged tissue for nutrients.
Yeah.
They don't have to have a specialized,
The hyenas, not the lions.
I like that very much. Thank you. So what is really exciting, and I want to spend a few minutes with this, is you spend time talking about what I'm going to call shock and awe microbiology. Okay. That is to say, things that are really exciting to hear. And I'm going to ask you a question about that in a second. And then you start talking about physical parameters to some degree. And I think that's really what's important. So let's first start. You were mentioning, we're talking about deep life, and you have a wonderful example in your book of an organism that lives in deep, deep mines and makes a living in an unusual fashion.
Mm-hmm. You talking about radiolysis of water?
I am.
Yeah. So Desulfurudus autoxviator, and probably other things as well. And this is not my discovery at all. By all appearances, seems to survive off of chemicals that are made from the slow radiolysis of water. So Earth is full of radioactive elements. This is from the primordial concretion of our planet. And so these things are slowly decaying over time. And as they decay spontaneously, which means radiation, the energy that's released from this radiation will split water into hydrogen and oxygen. And so and the oxygen is sometimes very short-lived, so you end up with the hydrogen. And so this hydrogen seems to be fueling life that persists very slowly, possibly over hundreds of millions of years or longer.
So it's really interesting to think about that. I have a colleague there's a mine in Michigan. He went quite deep and it gave me some banded iron formations from there. I have all kinds of toys on my thing. I love little trinkets. Do you happen to know Ken Nielsen?
Yeah. Yeah.
So I've known Ken for a long time, and one thing that always amazed me with Ken is he had a shelf full of curios.
Uh-huh.
And I don't know if he still does, but he would hand them to you and say, you know what this is? And I actually followed up on that. I have the same things.
Arguably, I took his job. He was retired. It was
sure.
But yeah.
But he will never truly retire.
I didn't, I don't have trinkets yet. I just moved in, so I have a couple books and a plant. That's all I got.
That's right. Well, no, I've got tons of stuff.
Better trinkets.
No, of course. But the thing is that he and I used to call it when I was talking with him, I would call it kind of a Zen approach to microbiology. Like, what does this mean? So I won't go into detail with, well, I'll show you. I have what was at one time a shell, and it's covered with manganese nodules.
Oh, that's amazing. That's beautiful.
Right. And this is from an anoxic estuary in Mexico, and a student picked it up for me and said, what is this? And it's manganese. And so those were, and we'll come back to that, breathing metal. And when I talk to folks, they get so fixated on the way that we make our living, where we take a high energy electron out of our food using NADH. And in fact, many microbes can steal electrons from other things, inorganic things like metals. Right. And when they do that, it changes their solubility, so they'll precipitate, or the reverse.
Or use the electrons directly, like Ken. Ken was absolutely instrumental in our knowledge of that.
Yeah, absolutely. So the point is, I think that there's a big drive to be narrow and specialized. And what I like about this is that you are not doing that. You're talking about all the different things. So with regard to some organisms that live quite deep, there was an expression that we used to use. I don't know if it's so old that you don't use it anymore. Slimes, subsurface, lithoautotrophic, microbial communities.
Yeah. I don't know why I don't use that word. I should look into that, but I do know that word.
Yeah. So the point is, and I'll put a link to that old paper in the show notes. When you dig deep, you'll find organisms that are using carbon dioxide that's been released from normal activity and hydrogen from, as you say, autolysis of water, and they're able to make a marginal living from that and grow deep, deep, deep.
I mean, there's other ways to get those chemicals too, other than radiation. Just water rock reactions like serpentinization will create basically everything you need to fuel life. Yeah. And I think maybe one reason why I probably should bring the word slimes back because it's a really nice word, but there was a debate back then about whether it was possible to have a truly independent hemolithoautotrophically based ecosystem. And this was when I was in getting my PhD, and I remember sort of getting the feeling that the consensus was you would never have it isolated from photosynthetic inputs. So like dead plant matter would always be getting into your system, and you can never separate the two out. And so it's always going to be a hypothetical asymptote. I think that is not, it can't be true. I mean, all the stuff that I'm looking at when we look at the carbon in these systems, we don't see signals of plant matter in our carbon isotopes. Mm-hmm. It's all chemolithoautotrophic. So I think the slimes were real. Like, there are these whole systems that could care less what's living and dying up here at the surface.
I agree completely. And, one of the things that I have to carefully teach students is that many of the enzymes that they learn are super important are actually microbial enzymes, nitrogenase and actually rubisco. Rubisco, that is one way that you can fix carbon dioxide into organic compounds. Nitrogenase, because actually dinitrogen gas is practically inert and you need to have something like ammonia that can be used for biological systems. So when I tell them there are many autotrophs, that is to say they use carbon dioxide for their input carbon. Number one, they have to build everything out of that, which is just amazing to me.
Yeah.
Secondly, they don't have to use light, but most students have only heard about rubisco being coupled to light.
Yeah.
And they get told that's what chloroplasts do, and they forget, of course, that chloroplasts were originally bacteria.
Right.
Right? Yeah. First evolved, last extinct. Yeah. So the point is, it's all about making a living. And I'll tell you, this is a little bit off topic, but I think you'll appreciate it. When I was in college and taking microbiology, I learned that standard aerobic respiration per input glucose gave you between 34 and 38 ATP. That was a big deal to me. Mm-hmm. Boy, that's great.
Yeah.
Glycolysis from glucose, you only get 2 net ATP. Mm-hmm. And then I take a pipette and I take some muck, some anaerobic muck, and I look at it under the microscope, and it's swarming with things moving around.
Things loving that 2 ATPs.
And it's reminding me that we have an assumption, the more energy available, the more life, and that's not necessarily so. And that's when I started to really think, are we looking at the world the right way? The majority of life on this planet is microbial, and quite a bit of it is subsurface. I don't know if you have an estimate of that.
Yeah, it's hard to do that subsurface comparison, because are you talking about number of cells, number of living cells, total biomass? And like, if you talk about total biomass, of course the plants win because of the trees. But if you look at a tree, the trunk is not living cells. If you just look at the living cells of a plant, it's actually very low biomass. Or do you count all the bodies? Do you count banded iron formations, which are basically the output of probably some precursor to Gallionella, this iron oxidizing organism, or Shewanella or something like that? I get twisted around in all these estimates. It's a lot. It's something like 10 to the 29 living cells in Earth's subsurface, which is 10,000 times more than the number of estimated stars in the universe.
There is a saying about Frank Sinatra, and you're thinking, what has this got to do with anything? Well, Frank Sinatra was a character, and someone was talking to his friend, Dean Martin, about something that Sinatra had done. And Dean Martin said, and I quote, "Friends, it's Frank's world. We just live in it." And I would argue it's a microbial world, and we just live in it.
Yeah. I mean, we just showed up like yesterday.
Yes. Yes. So I think a good thing to do now, if you wouldn't mind, is tell a adventure travel story, one of your favorites you can, about a place you went, the challenges you had, and what you found.
Oh, man. I guess
you have so many.
Well, yeah, there's always challenges. I think drilling in the Arctic was pretty challenging. It's just, you're so remote. You're remote, but at the same time, it's maybe not quite what you think, because it's not like we're, Roald Amundsen traveling for the first time with no resources. Like, we stay at a very comfortable research station in New-Ålesund and Svalbard. They do a fantastic job of providing our material and physical needs and food and stuff like that. But you go out every day into the field, and you have to deal with the polar bears, which are more numerous than people in Svalbard. And they're also getting a little weird with climate change. So you have to be kind of careful. They've been hunting on land more than they have. This is anecdotal. I'm not a polar bear ecologist, but they've been eating reindeer, which is new for them. They usually stick to seals, but people have been seeing them take some reindeer, which is seems a little desperate. So that causes, you want to be extra especially careful, because they seem to be on land more and they will hunt us. And so we have to be prepared for that. But then it's, the big problem are not really the polar bears. The big problem is this ice and rock and frozen soil that we have to drill through. And so we discovered on one of my first drilling expedition to Svalbard ever that our metal just didn't work. And so we were drilling. Everything was going great. And then we were breaking drill bits off the top of our drill rig. And we still don't really know why. It was just a bad batch of metal somewhere. But we just spent the entire time. There's nothing you can do. You can't order more drilling parts. You just have to weld some things together and hope for the best and make do with what you get. But we ended up getting pretty good recovery considering that we were breaking drill bits right and left. But, when that happens, you just go with it.
So it's always interesting to me when I hear about things that are found in I hate the term, but I'm going to use it, icy conditions, are how many of them are psychrophiles and how many are facultative with regard to that ability. And again, since you're not necessarily growing the items.
Right. We did some of these.
Did they have, and I see you already schooled me on the term optimum. Did they appear to grow better at colder temperatures?
They were really slow, and they did grow down to 10 degrees. We couldn't get anybody to grow faster at lower temperature. We just didn't get that. So we didn't get a true psychrophile because that's the definition of psychrophile. But what we did do is we looked at their extracellular enzyme hydrolysis rates. So just the background is that organic matter is big and microbes are small. And so getting your food inside that little body involves sending out extracellular enzymes to cut them up like little scissors and get the pieces back inside. So we used small substrate proxies and looked at the rate of degradation of the stuff around them. And we did find that some of them were putting out enzymes that were working better at lower temperatures. So it is possible that they were adapted to work with lower temperatures, but they didn't actually. The ones that we isolated did not grow faster at lower temperatures.
It's really interested me because it's pretty easy for me to have students isolate obligate thermophiles. I'm not talking about hyperthermophiles. And we used to do this routine. Yeah, and using very low nutrient media, and we would do that from hot water pipes. Presumably they're forming biofilms in there, and I 16S them in their thermos, and that's nice, right? Yeah. I've always been curious about the ones that live at cold temperatures, but I think we're running against that issue of time centrism, that they could be growing really well for the environment, but not my perceptions.
Well, and think about what permafrost is too. True permafrost never, what we think of as permafrost is sitting there's an active layer at the top that thaws in the spring and summer and then refreezes in the winter. But if you're below that, if you're in the permanently frozen part, you don't have a lot of liquid water around. So sometimes the limitations on those organisms are not about temperature. It's more about water activity.
So going from the cold, I know you have an interesting story about volcanoes.
I have a couple of interesting stories about volcanoes. What would you actually? I have a story I didn't put in the book. It just didn't really fit. Well, I don't know if it's a good story, but it was consequential. I did talk about climbing this volcano. It's Iraputuncu in Chile, and it was a gorgeous volcano. I mean, they're all beautiful. It's like picking your favorite volcano is like picking your favorite microbe. But Iraputuncu was very high altitude, and it was a very, very big challenge to get up this thing. And we did it. We made it up this place. I tell the story in the book about how we discovered that our respirators didn't work by going into the crater and getting gassed. It was kind of like getting tear gas or something. I had to run out of the crater, and I was scratching at my face to get this stuff off, snot running from my face. It was so anyway, we had some better masks, so we took turns with our better masks. But when I was going down the volcano, my colleague Jacopo Passati, who's an alpinist, he's an Italian alpinist, said the best way to get down a volcano is to jump. And he took a flying leap off the side of this volcano. I was like, he's not a risky guy. He's a trustworthy fellow. And I was like, what is he doing? This is awful. And then I looked, And it was gravel. It was like this, because volcanoes bust up rocks. So actually the
Right.
slopes of a volcano have no vegetation, and it's like just little gravelly rocks. And so he was planting his heels, like heel first, like skiing. He skied down this gravel volcano, and it looked like a lot of fun. So I followed him. And I jumped down this thing like I was Tigger, bouncing like on a bouncy castle. And I had so much fun. And I slid on my butt for a lot of it, totally out of control. I do have an image of this in the book. I'll send it to you. It was just pure joy. And when I got down to the end of this long slope and was dusting myself off and watching everybody else manage it, like, going for it and like diving down this thing, I realized that my phone fell out of my pocket somewhere halfway down. Why I left my phone in my pocket, I don't know. I mean, there was no signal up there, so I wasn't like on my phone, but it was in my pocket. And when I got to the bottom, it was not in my pocket. So my phone lives on Iraputunku volcano to this day.
And how do you take samples from there?
We took samples from the fumaroles. There's no water at the surface of the crater in Iraputunku. So it is not your best case scenario for finding life. But we had the opportunity. I was sort of, it wasn't a microbially driven site to visit, but I was there visiting with gas geovolcanologists who were like, oh, we're definitely going to sample the gases from the volcano. And so I collected basically precipitates forming at the mouth of one of the lower temperature fumaroles in the hopes of getting something. And we have not gotten anything yet. We haven't gotten any DNA out of it yet. So I don't know what we need to do. Maybe I need to go back, see if I can get my phone and get some more stuff.
No, I mean, that's a lovely story. And again, when most people think about being a microbiologist, they don't think of you butt skiing down volcanic slopes.
Losing my phone.
There you go. And there again is a cartoon that some of your students should create, No, I have to tell you on that subject, I really love it when my students do that, because I was talking about gliding motility, and I said that some bacteria secrete a surfactant in front of them, and so it's like their own slip and slide.
Yeah.
And then I had a student who did a cartoon of that and just turned it into me, which is wonderful.
Oh, nice. That's awesome. That's great.
So, what I'm thinking about now is one of the things that you talked about that's exciting to me are acidophiles and alkalinophiles. And I want listeners and viewers to know that pretty much protons are important to life. Having more protons on one side of a membrane than another is all hail Peter Mitchell, is pretty much the prerequisite that we need.
Yeah.
So if you're living under acidic conditions, the definition of pH is the concentration of those protons, those hydrogen ions, and the reverse is the alkaline conditions. So you talk about that in your book a fair amount.
Yeah.
And you might talk a little bit about what that means.
Well, if you think about if you're in an acidic pool, then you already have your chemiosmotic potential built. You don't have to push out protons. There's already, your protons are crashing down on you all the time, and they're like trying to get back in. So it's like, this has always bothered me. Or when I first started learning about this, I was like, well, shouldn't you just be able to breathe for free? You have a ready-made proton motive force. Just let them come in and kick your ATP synthase and turn it around and make some ATP out of it. Drive your flagella, use your transport, all the things that we couple to the movement of protons. But the problem is that it's too much of a good thing. To spend money, you need it to be controlled. It's kind of like trying to imagine spending money with $1 billion physical dollars in ones taped to your body somehow. Like, you can't walk around holding $1 billion in ones and actually make that be a good strategy for spending money. So
this is where Scrooge McDuck was wrong. Because he's in that swimming pool full of money, he would have drowned in that money, is what you're saying.
That's right. Yeah, there's problems. I mean, drinking out of a fire hose will kill you, even if you need water. So really what they do is that they just spend a ton of energy pushing the protons back out, even though they already have the gradient. they're constantly bailing. They're sending the protons out. They make themselves like a tank, protecting themselves against what the rest of the world is trying to bring in, is like loving bringing in. These guys are like, they have to like batten down the hatches to keep it from crashing down on them, which I think is just the fascinating way to live.
Now, I don't know if you'll agree with this, but I always thought if they were just letting in all the protons, that's going to acidify their cytoplasm. And most of our enzymes are pretty much pre-selected for near neutrality on one way or the other. So they would have to have enzymes that could really tolerate that kind of thing, as well as the rest of their innards, for lack of a better term.
Yeah.
And that's one thing that has always fascinated me about extremophiles in general, is what's inside is usually not that different from other organisms.
Yeah. Which is a little weird. And it makes you wonder whether life started in a highly acidic place, because it kind of looks like it didn't, because it's like they're adapting to it, and they keep their pH. I mean, from what you always have to have a caveat of, maybe we haven't discovered all of them yet. But the ones that we've discovered and worked on, their internal pH really doesn't dip below maybe 5 or so, which is already pretty acidic. Mm-hmm. But they don't go to 2 inside.
And then going the other direction, that's a lack of hydrogen ions. And I know some marine bacteria will use sodium ions for this purpose.
That's right. Yeah.
But again, there are limits. And I again, you have a good background in talking about the physics of all this. So if I misstate, hold me to truth. But I remember reading something by a physicist about the volume of E. coli. Mm-hmm. And at a pH above 10, there wouldn't be enough protons inside to run anything, just because it wouldn't fit, because there's too few. Now, I don't know if that's really true, but it is true that you can find bacteria that can tolerate very low pHs, but there's a limit to about 10, wouldn't you say, for alkalinophiles?
No, I would not say that.
What would you say?
12.
12? That's exciting.
12.7.
Wow. 14's highest.
And I know the name of it too.
Go.
Actually, this is Ken Nielsen's work. It's going to be a great name. It's going to be very evocative. NPL UPA2.
NPL
UPA2.
UPA2.
It's one.
Is this from one of his serpentization sites?
Yeah. So it's from the Cedars in Northern California.
Yes.
But people have found it in other places as well.
That's amazing.
It's an uncultured phylum level bacteria. It appears to be a bacterium. When we look at these things in like the deep branches, it's like, I don't know, are you really bacteria? It seems to be an autotroph that has a Wood-Lungdahl pathway for autotrophy, which, is distinct from the Calvin-Benson-Bassam cycle that we learn a lot about with plants. Wood-Lungdahl is something sort of maybe more primordial that a lot of microbes have. So they have one of those, but their acetyl-CoA synthetase, that is one of the key enzymes for Wood-Lungdahl, is kind of like, it looks like a natural chimera of an archaeal version and a bacterial version. And to have something so central be cross-domain is a little uncomfortable and weird. And if only one group had found it, then we would say that's a bioinformatic problem. You created a chimera in your computer, you created it when you were sequencing it, when you were amplifying it, something like that. But other people have pulled it out of other places as well. And so it seems to be a natural feature.
Yeah, I don't believe data unless I see it 3 times. I mean, it really needs to be replicated. And it's a good thing for folks to remember. And I'm not trying to claim that there are a lot of errors. There are people that are doing that now and talking about science in general. But what I am saying, when you look especially at low abundance sites like this, you can get unusual types of results via bioinformatics, and you have to confirm them.
You do.
And it's hard because sometimes it's very exciting.
Well, one of the really cool things about bioinformatics is that you get these long strings of DNA that are hard to recreate randomly. So if you get that same long strand of DNA in the Pacific Ocean underneath in a subsurface mud volcano, and then by one lab, and then a different lab in the Cedars in Northern California pulls out that same stretch of DNA or a very similar stretch of DNA, you didn't get that by accident. You can feel very secure. So it's an extraordinary finding that has rolled up in it an extraordinary support.
So I guess I'm watching our time carefully. And I do want you to spend a couple of minutes talking about, and I'm going to actually change my deadly centrisms because of you. I am going to, and I'm going to try and see if I can put some Latin in there for time centric, because you do spend some time in your book talking about things that appear to have a replication time of, I'm just going to say very long periods.
30 years.
Yeah. And that's amazing because most people think, and this always kind of irritated me. They said, E. coli's every 20 minutes, but that kind of depends on conditions and all the rest.
Persister cells.
Yeah. But, oh yes, persisters really bug me too. But what you're saying is there are many microbes. And again, it's like when you talk about the different phases of growth, and you'll say, where do you think you find most living microbes? Do you find them in logarithmic phase or in stationary phase? And the truth is, a lot of them are going to be in stationary phase.
Really? There's a higher percentage of living microbes during stationary phase than in log?
No.
Okay.
But they're very persistent. They're constantly evolving. This is what Steve Finkel is. Yeah. I want him on this podcast to talk about.
I'll put in a good word for you. He's great. Yeah. His work has really influenced me quite a bit. And he studies E. coli that he basically gives food to once and then waits for years. And
yes,
the dynamics of what's happening. I want to acknowledge precedent here and say that this has been done many, many times in the past by people who've sort of left their cultures sit out with no new food. But Steve's taking it one step further and actually studying what's happening inside that tube. And what he finds is that there's a lot of growth, there's a lot of dynamics, that they can really eke out a good living over a very long time. Of course, this is all E. coli. So that's what E. coli does.
Sure. And again, that's interesting because it makes you wonder, where do you find most microbes? And we have all these assumptions about things, and they're generally wrong because we don't have, I don't want to say a psychology of microbes because that makes people roll their eyes at me more than they generally do, I think. But what I would say about this is that we don't have a microbial perspective. And it's hard to have one because the microbes that we focus on are things that tend to cause disease or do things that we care about in one way or another. And what I like about the kind of work that you do is you're looking at what's there. And so this idea of some microbes reproducing very, very slowly may reflect an awful lot more of life on this planet than we think.
It could. I mean, if everything was growing in log phase like E. coli, we'd just be smothered.
Yeah. And so that's a fascinating idea to me. And I really like the idea. On a previous episode of this podcast, we were talking with one guest a little bit about the Viking landers on Mars and how what essentially they did, and again, I mean, no disrespect to NASA, is dump a bunch of fluid, water, nutrients on a sample of soil. And from that, they determined there were no living things there. And our guest thought perhaps they should have simply gently humidified the sample. And that sounded reasonable, but after listening to your time centrism idea, maybe they should have watched it for a long period of time. Do you have thoughts?
Yeah, absolutely. I mean, we see that, you must know from culturing, like when you put things in a culture medium, sometimes you have to wait for a very long time to see anything. It's rare for them to immediately respond, even if they are sort of a normal, fast-growing organism. So if you want to find the ionophiles, that's what I'm calling these things that by nature live for, it's a different word in the version that you have. I had to change the name for Google reasons. If you want to find an ionophile, these, by nature, they have to grow very, very slowly. These organisms, you would have to wait a very, very long time. And I would also add that I don't think you can ever conclude a negative result because we haven't tried everything.
I think that's well stated. It's always, I sometimes want to say to the people who are negative about it, what would be positive evidence to you?
Yeah.
And then they need to say to me, what would be negative evidence to you? Right?
And that's hard to fully say. Absolutely.
Agreed. I wanted to say now, I strongly recommend, Yeah, I am going to make a plug because I really think this is worthwhile. Anyone who likes this podcast, I want them to get this book. I mean, I got to tell you, I'm not being paid to say that. nor am I getting my copy is not signed. So but I'm going to work on that with you. And it's a really interesting exploration of the depth and breadth of the microbial world in a way that in one volume, and it's accessible to lots of different people. That's the point that I think is important here. Yeah. Is that, if you're more interested in travel logs, there are travel logs. By the way, I love your bit when you're in the deep sea submersible because I get so claustrophobic every time I read those things. Right? First time I saw Alvin, I go, I am not getting in that thing. Yeah.
No, it's great. I love it. I'm very comfortable in small spaces.
Yeah. Ditto with caves.
Yeah.
I mean, lots of stuff. One thing I wanted to mention, and this is a little bit random, but it's something that fascinates me. Were you aware of some types of cyanobacteria that are able to get some photosynthetic energy out of the triboluminescence that happens when hot water hits cold water around hydrothermal vents?
Yeah, Bob Blankenship's work. Yeah. It's pretty cool.
It is. And people say things like photosynthesis had to happen in response to the sun. And I'm thinking, maybe.
Yeah, that's right.
Right?
Yeah.
But that's what I love about microbiology. That's what I love. And this level is really, I showed it again, didn't I? This level of it is really important to me. And it's very inspirational. I want to thank you for writing it. I can tell it was a labor of microbial love.
Yeah. It was, I'll tell you, it was just whatever was on my mind. I didn't do much research for it other than just the research that I do. It sort of is the culmination of just what I have lived and what I've studied in my life. Mm-hmm. So for that reason, it was just fun to try to think of how I can best give this cool stuff that, like, I feel like it's just like bursting out of all of our heads, those of us who work on this. I'm like, everyone should have this. I should give this to them.
You'll laugh at this, and this will be part of your gift. I have two expressions that I use with my micro students. One is hashtag OMG.
Hashtag what?
Hashtag OMG, okay, which stands for overwhelming microbial greatness.
Okay.
Nice. And the other one is hashtag WTM, like, what the microbe?
Nice.
And this is what is exciting to me, to hear this kind of things. Too many times microbiology is taught, and I'm not pointing fingers and I am not making fun of anybody, like a bug parade. Here's this organism. It does that. I want the vitality of the microbial world and the excitement of the microbial world to be front and center. And this is a good example of it.
Cool. Thank you.
I think for the last thing that I wanted to talk about is, would you mind telling listeners and viewers about the path that took you to the microbial sciences?
I grew up on the coast of North Carolina, in Beaufort, North Carolina. And so I basically did a ton of, whenever the local universities in North Carolina have marine stations there, whenever they did outreach, I signed up for everything that I could. And so that sort of introduced me to the oceans and the microbes that are present in the oceans. But I didn't really latch on to microbes per se, beyond like just chemistry and the whales and dolphins and stuff, until I did a summer program at North Carolina Central University in Durham, North Carolina. And there was a professor who was nearing retirement at the time, John Mayfield, who just was like, there is a hidden world of microbes. They are everywhere, and they are doing the most important things that we need for the world to happen, totally silently, invisibly, and it just absolutely opened my mind. And I thought, I gotta work on this. But then when I went to college, I kind of fell into chemistry because I couldn't not. It's too good. Like, chemistry is fascinating. And so I had to major in biochemistry just because that was great. But I was always headed back to microbiology.
It's funny. I fell in love with genetics early on. And I explained the difference between biochemists and geneticists as follows, is that a biochemist looks at a black box, that black box being a cell, and they drop a bomb on it and look at the pieces.
Huh.
And geneticists look at that black box and they look at what goes in and what comes out and try and hypothesize what takes place inside.
Mm-hmm. Mm-hmm.
And the exciting part about today is that those approaches have merged to some degree. And that's what I really like. So many times I will hear from students the only path to the microbial sciences is doing X, say like health-related materials. And I'm not upset with that if people are interested in that. What I want people on the podcast to understand are there are lots of different ways that you enter the microbial sciences.
Absolutely.
Yeah. And once again, just like with many people, myself included, some of those additional courses that take place that you wouldn't think, oh, I shouldn't apply for that. You should.
I would say just do whatever's got a good teacher.
Yeah. Yeah.
And that's kind of what I did with chemistry. I just had such fantastic professors at Swarthmore College where I went to school. I was like, okay, I'm going to stick with this and learn from these people. And I think it was the right choice.
There's a lot of hate that sometimes my pre-health students put on chemistry. And I tell them, if this is the hardest thing you ever have to do as a physician, that's pretty lucky. Right?
Yeah.
Well, Karen, I have really enjoyed chatting with you today about this.
Yeah, sure.
And I've learned a lot. And I want to be clear to everyone, there are some things that I was wrong about. And Karen, Dr. Lloyd, very carefully schooled me. And so now I know more and I can bring that to my classes. And I want to thank you for that.
Oh, sure. Yeah.
So I want to wish you and yours the very best. Congratulations on your new position at USC. Yeah. Even though I got my undergraduate degree from UCLA, I still salute you. Yeah. And I wish you every bit of good luck.
Thank you so much. And thank you for having me on here, and the best to you and yours.
Thank you so much. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with, as usual, awesome links well worth your time, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Karen Lloyd is a professor of earth science at the University of Southern California in Los Angeles, California. Many thanks, as always, to David Renata for superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our quality quorum today. See you next time on Matters Microbial.